G'day, my name's Jordan Hansford. I'm one of the oncologists, neuro-oncologists at the Royal Children's Hospital. And I'm your MC for today's online edition of the VCCC's Monday Lunch Live. I'd like to acknowledge the traditional custodians of the lands on which myself and today's audience is located. I'd also like to pay respect to elders both past and present, and extend that respect to other Indigenous Australians present online. We have some very interesting speakers coming up, so please continue to register your interest on the VCCC websites. We'll also have a learning page if you'd like to see the previous topics that have been presented. If you have any questions for today's presenters, you can do this via the Zoom and the Q&A function. You can also raise your virtual hand during this time if you'd like to speak to the panel. Please submit your questions throughout the presentation and we'll aim to answer your questions at the end of the session. It's now my distinct honor to present Associate Professor Misty Jenkins, a friend and colleague, collaborator, who works at the Walter and Eliza Hall Institute. Associate Professor Misty Jenkins is going to talk to us about novel CAR T therapy for adult and pediatric glioma. Misty is an NH and MRC fellow and lab head in the immunology division at Walter and Eliza Hall Institute for Medical Research, where she researches cellular immunology and cancer immunotherapy. Misty completed her PhD in immunology at the University of Melbourne, followed by postdoctoral positions at the Universities of Cambridge and Oxford, and of course, Peter MacCallum Cancer Center in Melbourne. Misty has a long-standing interest in CD8 T cells cytotoxicity and the immune synapse. In her current research program, researches the use of immunotherapy for brain cancer. Misty was awarded the L'Oreal for Women in Science Fellowship in 2013, the Tall Poppy of the year 2015, was awarded the Westpac Australian Financial Review Top 100 Women of Influence in 2016. In addition to her research career, she is experienced in governance and is a board director for Monash Health and co-chair of the Indigenous Health Medical Research Future Fund. And I will hand it over to Misty, as I am certainly looking forward to this chat.
Thanks, Jordan. Can you hear me okay?
Yeah, we can.
And can you see my screen okay?
Yes.
Okay, perfect. Okay, oh, I'm so excited to be here. I love Peter Mac, so I'm just really excited to tell you about what we've been up to. I want to also start by acknowledging the traditional custodians of the unceded lands on which we live and work. In my case here in Melbourne, it's the Wurundjeri people of the Kulin Nation. And for those of you dialing in, we acknowledge those elders past, present and emerging, and acknowledge the continued connection to land, water and community. Thank you so much for having me. It's a real privilege to come back and tell you what I've been up to, some of the things that I've been up to over the last 4 years here at WeHi. Today I'm here to talk about T cells and really how we can manipulate them to use as a living therapy in the fight against cancer. And, you know, T cells perform a variety of critical functions in the body, as shown here, this beautiful lattice light sheet of the T cell migrating around, recognizing antigens, killing targets, mainly via apoptosis, although other death mechanisms as well, releasing cytokine to further augment the immune response, recruit their army, and as shown here in this gorgeous video, the orange T cell ultimately delivering the kiss of death to the cancerous target cell. And so this lethal hit is directed by the polarization of the centrosome, shown here in red in this movie on the left, which directs the granules to dock and fuse at the plasma membrane. And in this movie on the right, you can see the granules in red following the blue centrosome, and I'm not sure if you can see my cursor here, and where they secrete their contents into the synaptic cleft, including proteins such as granzymes and perforin. So being at WeHi, it would be remiss of me not to mention the role of Brunette in recognising the important role that the immune system plays in detecting and destroying tumour cells, because Brunette and Thomas back in the 1950s extended their theory that the immune system has evolved to keep us safe from pathogens to also describing cancer as a failed immune surveillance. However, it really took another 50 years for this to gain acceptance, you know, when mice lacking effector molecules such as interferon gamma and perforin developed cancer. Of course, this was quickly followed by a number of landmark papers including Jim Allison's work, an anti-tumour immunity checkpoint inhibitor blockade. And really, immunotherapy has become the largest advancement for cancer since chemotherapy really took the world by storm. I think it's no surprise now that we're treating patients with personalised medicine, and I still think that we are in the infancy of what immunotherapy can do, and not just for cancer, but other diseases as well. So how do T cells recognise their antigen, and how can we genetically manipulate those recognition receptors? So this schematic on the left outlines the T cell receptor, which is a heterodimer with its alpha and beta chain, which non-covalently associates with the CD3 complex. And this is really essential to provide the molecular and the kinase scaffold required for signaling and function. And co-stimulation is provided in the form of these proteins shown here. And so really the immune sensor called ICAR, chimeric antigen receptors schematic shown on the right, and originally made from monoclonal antibodies in constant regions of the T cell receptor. But Escher in the lab of Steve Rosenberg modified this design to include an ectodomain from a single chain variable fragment. And I'm going to refer to this throughout the talks as scFv. With a heavy and light chain of an monoclonal antibody, transmembrane domain, and endodomain with a signaling from the CD3 zeta. Now, most CARs subsequently designed have followed basically the same structural pattern with the incorporation of various co-signaling endodomains. It's important to keep in mind, though, that one of the main points of difference is that in the periphery, T cells don't normally receive signal 2 or co-stimulation every time they see antigen. And this is relevant for the choice of co-stimulation when designing CAR T cells. And really, CAR design could be really a whole seminar in itself. But today, I really want to present our work designing new CAR T cells that target brain cancer. But I should also point out that these antibody binding domains, or these scFvs, have also been adopted into additional biologics. So these include BiTEs, bispecific T cell engagers, which are essentially like a double-headed antibody fused to an anti-CD3 scFv to engage the T cell and bring them in contact with the tumours. And we're also working with a company in the US called TCR², who have a proprietary linker called a TRUC, which incorporates the scFv into the native T cell receptor by tethering it to a various CD3 subunits. I'm not going to go into depth about this today, but these designs really are just the beginning, and many of you in this audience will be very aware of the further genetic lodging gating strategies currently being employed, such as notched in CARs and inducible secretion of BiTEs into the tumour microenvironment, as well as other engineering strategies such as suicide switches. But as I said, that would really be another talk in itself. So of course, I think the success and approach of these sort of immunotherapies are absolutely dependent on the tumour indication. And I think there can be no assumption that an approach that works well in blood cancers or lung cancer may also be able to be applied in a delicate location like the brain. And so particularly when taking into account trafficking across the blood-brain barrier, limiting inflammatory cytokine secretion, and tackling immunosuppressive environments. So this schematic provides an overview of the process, which will be familiar to most of you in the audience today, in which blood is harvested from the patient, activated with anti-CD3 and anti-CD28, transduced with viral vectors usually containing the CAR, although again, lots of different strategies are being employed around this. The cells are proliferated in culture and then returned to the patient. And really the take-home message of this slide is that influencing tumour aggression, but importantly, influencing T cell fate can be manipulated at every single step along this pathway, right from the phenotype of the T cells, which form the starting population here, how they're activated, under which conditions, which cytokine regimes, CAR design, which is another talk, and also the reinfusion protocols, which includes the dose ratios and preconditioning regimes. T cell therapies are typically administered as a defined number of cells per kilogram of body weight, but also, of course, T cells will expand and replicate after transfer. And so the dose doesn't necessarily resemble the final steady state number, which can vary among patients. And preclinical studies have shown that, improvement in tumour regression, but with increased numbers of dose cells transferred, but also an increase in side effects. And so, the ability of the cells to proliferate and persist might be more important than the initial dose, which is why we think that the way that they're initially programmed is very important. And we have a number of projects underway examining this area, but won't be the focus of my talk today. So this therapy, of course, has had its successes and also its pitfalls. And this is Emily Whitehead, who was the first paediatric patient to be treated with anti-19 CAR T cells to treat her refractory leukemia. She's now 8 years cancer-free and doing really well. So when I started my lab here at WeHo, I really wanted to ask the question, how can we take the success stories like Emily's and translate it into some trickier cancers like brain cancer? Brain cancer kills more children than any other disease and really has quite a dismal survival rate, as shown here in these statistics when you compare it to other cancers. And in fact, if you look at a high-grade glioma like paediatric GBMs or DIPG, a diffuse intrinsic pontine glioma, and this, I should point out, this is being reclassified to diffuse midline glioma or DMG, we can see some of the most devastating brain tumours with no effective therapy, and they're universally fatal. So this tumour presents in kids aged between 5 and 10 years old. The tumour grows in and around the nerves, just in the brainstem, so it's unable to be surgically removed. And radiation therapy can give these kids a few extra weeks or short months, but really from the time of diagnosis, the average survival time is between 9 and 12 months. And as I said, is overwhelmingly fatal. Now, there can be no mistake that it's worth pointing out that adult and paediatric brain tumours are actually very different diseases. In the adult setting, glioblastoma is the most common brain tumour, as shown here in blue, however, is extremely aggressive and displays the poorest survival, as shown here in the purple diamonds. It's highly malignant and exhibits widespread infiltration and is very resistant to therapy. The current standard of care for newly diagnosed glioblastoma patients includes surgery, temozolomide, combined with radiotherapy and alternating electric field therapy. But after recurrence, there's no standard therapy and survival's less than 9 months. So in my lab, we decided to take on this challenge and to designing new CAR T cells to treat high-grade gliomas in both adults and children. So what's been done so far? And well, actually not very much as yet. There's just a one slide summary of the first 4 of the first in-human CAR T cell trials in the brain. One patient treated intracranially with a CAR against IL-13 receptor alpha-2, also targeting HER2 and EGFRvIII have been reported. These trials have mostly been in adults with the exception of the HER2 specific CAR T cells and 7 paediatric patients. In some of these trials, a T cell trafficking to the CNS was examined, engraftment, persistence, and of course the CAR T cell response. And really, to summarize this literature in a few seconds, the take-home message is that the T cells were shown to effectively traffic into the brain and were shown to be safe, but that antigen escape is really a continuing problem for these heterogeneous tumours. There are some caveats to these studies, however, with the V3 trial, waiting for patients to relapse before administering the CAR T cell therapy, but they never re-investigated the V3 status of the tumour. So it was probably no great surprise that the patients relapsed with V3 negative clones. So, so knowing what to target is absolutely key. And, you know, proteins are drug targets and cell surface expressed proteins are CAR T cell targets. And despite the large number of CAR T clinical trials, only Chimeric and Yescarta targeting CD19, shown here in the blue arrow, have been FDA approved, and there are still relatively few target antigens that have been identified across tumour indications. I've highlighted in green the targets that have been identified in the brain, and also you'll see by the y-axis the small number of ongoing trials. So it's still really early days. But we also know that for various reasons, you know, I think a monotherapy approach is unlikely to yield positive outcomes, and particularly in like a solid tumour setting, therefore the identification of novel targets are really urgently required. So this brings me to the approach in my lab. We're currently working out known and validated targets. So sorry, that just clicked over. We're currently working up known and validated targets. We're also generating our own clones of scFvs to target them with CARs and other biologics, as well as we've implemented a whole discovery platform to identify new and novel targets. And I'll talk a bit about this now. We're also deeply examining combinations of different T cell activation protocols to generate effective immunotherapies, but that'll be the focus of a future talk. Okay, so the discovery. So we take a two-pronged discovery approach. We access publicly available RNAseq data sets from primary brain tumours and screen for transmembrane domains, as shown in this schematic here on the left. So you can predict which proteins might be present at a high level on the tumour cell surface. And we do this work in collaboration with Melissa Davis and her team at WEHI. We also use cell surface proteomics to directly identify using mass spec which proteins are present at the cell surface of primary tumours. This approach takes advantage of the fact that most cell surface proteins are glycosylated, which we label with a biotin tag, and before lysing the cells and pulling down the biotin labeled protein. So once the beads are removed, we can use mass spec then to identify peptide fragments and gives an indication of expression level. And we do this work in collaboration with Jared Sandow and Andrew Webb's lab, Proteomics lab at WEHI, with the aim, of course, to identify novel targets. So this approach has successfully enabled us to identify new targets, and I'm just showing you a heat map of the cell surface expressed proteins identified in both primary brain and also patient-derived xenograft lines, both from adults and children. And the first thing really to point out is that, and I think, you know, most of you will know this, but as soon as tumour cells are removed from their normal environment and placed in culture, they change. And so, you know, as shown here, the heat map comparing cell lines of primary tumours, and I think this highlights the caveats. You know, when using cell lines for antigen discovery, they're really quite removed from primary tumours, particularly in the brain. And also adult and paediatric tumours are also very different diseases, and this was also highlighted in the proteomic analysis, as you can see here. So we've got ongoing collaborations with Kate Drummond and Andrew Morikoff and Jordan Jones at the Royal Melbourne Hospital, where we access primary glioblastoma tumours fresh out of surgery and are currently analysing this data. But however, I did want to show you this graph here on the right, a principal component analysis of 2 primary paediatric DIPG tumours, which are extracted after rapid autopsy following the patient death. And as you can see, importantly, the patients separate normal from tumour on PC1, further providing promise that the clustering will enable identification of interesting targets as the stratification is more pronounced between disease and healthy. As you'll appreciate, it's pretty difficult to obtain samples of healthy brain. No one really wants to give you a chunk of their healthy brain. So these matching samples of healthy are from this matched from the same patient taken during autopsy from regions of the brain that are very distinct from the tumour in the pons or the brain stem. These are really precious samples, and we're so, of course, very grateful to families who make the generous donation to, you know, decision to participate in research so that we can hopefully use these tumours to help others. And we do this work in collaboration with Jordan Hansford and our collaborators at the Royal Children's Hospital, as well as the Hudson. And so really just to show that this approach is working, our pipeline is established. We've identify the hits that come up from our tumours are commonly, we have identified some commonly known glioma antigens such as IL-13 receptor alpha-2, EGFR, HER2 and EphA2 all came up as hits on this screen, which was really great to see. And hopefully to have, you know, a future talk, I can come back and tell you about some of the work of some of the novel ones. But you would have seen that, so HER2 also did come up in DIPG, and we propose this could be a good target for immunotherapy. And we have labelled various DIPG cells with anti-HER2, and these found that these ones here are quite high, made CAR T cells and did a killing assay and were able to kill them. Here's showing you high HER2 expression here from the proteomic analysis, and just very, very preliminary hot off the press data. We are now validating this in vivo and seeing some responses in mouse brains. So now I want to tell you about the novel scFvs that we've made for known antigen targets in the brain. The first is EGFR and EGFRvIII. EGFR has been studied extensively and contains a large extracellular module. It exists in 2 conformational states. It's either tethered or extended, and the 4 extracellular domains sort of curl in on each other as shown here in the schematic and create a structure called the dimerization arm. And many cancers actually overexpress EGFR, and EGFR can become untethered and extended. This causes oncogenesis, and it also means that EGFR has been a good therapeutic target. And more than 30% of glioblastoma tumours actually also contain a mutation in EGFR called V3. This is a 801 base pair deletion of exons 2 to 7, and it causes the receptor to be constitutively active. And so what's beautiful about this is that it's exquisitely tumour-specific mutation. So it's not found in any healthy cells. And in addition, V3 mutations have also been found in 40% of paediatric DIPG and 17% of paediatric high-grade gliomas as well. So we thought this was a good target to make a novel scFv to. And so in fact, there was a chimeric monoclonal antibody made against amplified and untethered EGFR, made many years ago now, actually, at the Ludwig Institute. And over the years, there have been many studies and trials targeting EGFR via antibodies. And the current clinical lead is known as 806, which recognises a cryptic epitope exposed when the receptor is in a transient state between active and inactive dimer. And so the V3 mutation exposes, as you can see, exposes the cryptic epitope and then allows binding, and it's currently in several clinical trials. So we worked in collaboration with a Melbourne biotech company called Affinity Bio that have a proprietary retained display screening platform to generate EGFRvIII specific scFvs. And so this project has been led by Ryan Cross, a senior postdoc in my lab, and also the talented PhD student, Beck Abbott. And so in collaboration with Lucy Sullivan over at the Doherty, we performed SPR analysis and showed that these novel scFvs were of very high affinity. And in fact, they're more than 300-fold higher than the Novartis EGFRvIII specific clone called 2173. And you'll see that I use this as a comparator in my studies. And once we had confirmed the affinity and specificity of our clones, we confirmed that the recombinant scFv could bind V3 specifically on our target cells. This is some glioblastoma target cells here, looking at flow cytometry plots of recombinant antibody binding to V3. So that was great. And then we used the positive control down at the bottom as cetuximab. And we subsequently went ahead and cloned these scFvs into a second generation CAR with a CD28 co-stim tail, and then retrovirally transduced primary T cells, and then determined their cell surface expression. And we consistently get really good rates of transduction efficiencies in primary T cells, shown here in these representative flow plots. And I'm just showing the C2 CAR T cells, so that's clone 2, C2 CAR T cells, that's our endogenous V3 specific CAR, contain an endogenous MIC tag, as shown here on the y-axis, and then Novartis comparator 2173 clone contains a flag tag, and there's an M-cherry intracellular reporter, and we get good concordance of protein expression in both CD4 and CD8 T cells. So next we performed a standard killing assay to show that our CAR T cells were functional against V3 expressing glioblastoma targets, and you can see that they killed well, with the negative control here being an M-cherry empty vector, empty CAR vector control. And importantly, our C2 CAR T cells killed equivalently to the Novartis clone. And in fact, this movie just on the right also shows live cell imaging of our novel CAR T cells expressing the M-cherry reporter, as you can sort of see in pink, and then also labeled with fluo4 calcium indicator that flashes green when it recognizes the target. So this movie also shows specificity of killing, but also that the T cells do induce a morphologically distinct apoptotic cell death. So we next examined the secreted cytokine profile of the C2 CARs compared to the 2173 clone and compared to empty vector. So we had co-incubated cultures together with either the parental or the V3 expressing glioblastoma lines, and used a standard cytokine bead array. And typically concentrations were equivalent, and we used as a positive control here, we've used CD3 activated T cells, and then activated through the CAR receptor specifically. And really overall, you can see that whilst the C2 CD8 CAR T cells displayed enhanced secretion of gamma compared to the Novartis clone, actually across the board they secreted significantly less cytokine and chemokine. And I'm just showing you here TNF alpha, and strikingly the MIP1 alpha bucket loads less MIP1 alpha than the 2173 CAR T cells. And the CD4 CAR T cells also showed a reciprocal response, and with a reduction in interferon gamma secretion. And critically, I think, importantly, there was a negligible cytokine secretion when they were co-cultured with the parental line, reaffirming a lack of cross-reactivity. We examined a whole bunch of cytokines and chemokines, such as IL-6, IL-4, 5, 10, they were not detected. So we next evaluated the C2 and the 2173 CAR T cells for the expression of the immune checkpoint inhibitor PD-1, and when co-incubated with the V3 targets, the C2173s displayed a greater percentage and higher level of expression as determined by MFI, as you can see here, of PD-1, whilst an increase in PD-1 was observed in both. It was a lesser degree than that induced by the anti-CD3 co-stimulation. So that was good to see. So next we, after the in vitro characterization, we established intracranial tumour models to evaluate their function in vivo. So we labelled the glioblastoma cells with the firefly luciferase to allow monitoring of tumour growth. These human cells are implanted into NSG mice, and then we deliver the tumour cells intracranially via a slow osmotic pump. It delivers 5 microliters over 3 minutes. So it's tiny, tiny amounts, and this ensures no backflow of the tumour cells and into other regions of the brain. And we do this, as I'm just showing you here on this picture on the left here, at a location 2 millimeters lateral to the bregma at the coronal suture and 3 millimeters deep. And we're also doing this now 2 millimeters deep as well. And then we close up the hole with bone wax and suture the mice shut, and actually the mice recover very well from this procedure. So we established tumours in these mice over 7 days, image them, randomly distribute them to a treatment group, and then mice received 1 single intravenous dose of day 8 post-activated CAR T cells. And then the mice were imaged weekly by luminescence, and in vivo tumour cell growth was quantified in response to treatment. And as expected, this is quite striking data. I think we're really excited to see this. As expected and in accordance with previous studies, as shown on the left, mice treated with the empty vector CAR T cells failed to control tumour growth, as shown by this bioluminescence in the brains of mice. And in contrast, strikingly, 1 week after the single infusion of the CAR T cells, tumours in mice treated with both C2 and 2173 T cells, both of our clones of CAR T cells, as well as here the Novartis comparator, were below the limit of detection, and this remained stable for the following month. So this data really nicely shows that our novel CAR T cells can mediate rapid and complete clearance of intracranial EGFRvIII expressing tumours. And just because this data was so striking, here's the repeat experiment with it quantified by BLI. We've done that 3 times now, and just nearly ready to submit this paper. But also strikingly, that the histologic examination of H&E stained sections of the brain show that there was visible tumour present with morphology consistent with high-grade glioma. So this empty vector brain slice you can see on the left, this huge sort of purple mass of cells is all tumour. And you can see in these treated mice there, we just couldn't detect, and we had these looked at by a neuropathologist, and I should have Colleen Darcy's name on this slide at the Royal Children's Hospital, who did this work for us. And no tumour cells were present. You can see in the small inset boxes on the right, we also labelled the brain sections with antibodies to CD4 and CD8 to detect the T cells in brains of mice treated with the CAR T cells. And you can see that they were visible and were detected. And this was actually a full month after injection, and certainly at least 3 weeks after tumour clearance. So that indicated, and they were also, I should also point out that as you can see, that they're mainly in the periventricular white matter and the choroid plexus, and indicates effective trafficking across the blood-brain barrier and into the brain. So interestingly, despite administering, and I know that lots of you at Peter Mac have seen this as well, administering a 1-to-1 CD4 to CD8 CAR T cell ratio, the flow cytometric analysis of organs from CAR T cells after T cell transfer, again 3 weeks after transfer, revealed a significant proportion of CD4 CAR T cells in both the blood and spleen compared to the 50-50 in pre-infusion ratio, suggesting that either the CD4s have survived or proliferated better, and that is true also, as you can see here in the brain as well. So pre-infusion, the CAR T cells retained a 44 positive, 62 positive phenotype, low expression of PD-1, uniformly positive for granzyme B. However, 3 weeks after transfer, flow analysis identified 4 and 8 T cells harvested in the spleens, lymph node, blood, and the brain, as shown here, and determined changes to phenotype. And majority exhibited a central memory phenotype prior to infusion, but then afterwards predominantly differentiated into effector memory phenotype. And you can see, in fact, the highest proportion of effector memory cells, effector cells, are in the brain, not surprisingly. Now, I just want to show you just a couple of sort of teaser slides, really, because we're quite excited that one of our clones, that both of these clones are EGFRvIII specific, but one of them also binds untethered EGFR as well as EGFRvIII. And misfolded EGFR. So acting like that 806 antibody that I mentioned earlier from the Ludwig. And these flow cytometry plots show our recombinant scFv binding cell lines known to overexpress EGFR, both in glioblastoma and astrocytoma cells, as well as lung, colon, and breast. And so given the propensity for this C1 CAR T cell clone to bind untethered EGFR, we tested its ability to kill glioma in this is an IncuCyte kinetic killing assay, and you can see here really nice binding, but also really nice function and killing against EGFRvIII negative, but EGFR untethered expressing glioma cells. And this is, I should mention out, this is all work done by Ryan Cross. And in addition, we tested other indications, just shown here briefly, for lung, breast, and colorectal cancer, and also showed binding and killing against these lines, expressing the untethered forms of EGFR. So in very new, hot off the press work, we've also further tested these CAR T cells in vivo in models of lung cancer, and both subcutaneous tumours, but also the secondary brain tumours, as they often metastasize to the brain, and provided evidence here of tumour-specific efficacy as well. So whilst it's exciting to think that we've set out to generate these therapies for brain, there's also likely to be other indications that may benefit, and I'd be really interested to talk to some of you in the audience that already have established models in these areas, and if you want to chat about it, get in touch. So in summary, we have de novo generated 2 very high affinity novel tumour-specific EGFRvIII scFvs for glioblastoma, and also we have these as CARs and also as BiTEs. We see very high cell surface expression in vitro killing. We see striking regression of intracranial tumours with both of our novel clones of CAR T cells, but also one of these clones also binds and kills overexpressed EGFR. And I've got an asterisk here because we're just still working on doing the epitope mapping to work out exactly where, which residues this scFv is binding to on the untethered form of EGFR. And how am I going for time? Good. So next, I just wanted to talk about our work in paediatric DIPG. And because in 2012, a key breakthrough for DMG was discovered by several groups who discovered a key mutation in histone 3.3 and in 80% of these tumours. And so this results in a replacement of lysine by methionine. And also this mutation actually confers a clinically and biologically distinct subgroups. And these patients with this mutation have an even worse outcome and shorter survival. K27M is a powerful driver of disease. It can't be downregulated by the tumour cell, and it remains, it actually results in it like a global hypomethylation of the repressor complex and enhanced gene expression. Also the methionine residue actually serves then as an anchor residue, and it's able to bind and be presented by HLA-A2. So this makes it a really good target for CAR T cell therapy. So I'm going to now debunk the fact that CARs, Ts can only recognize proteins. They can also recognize peptide-MHC, which means that we can now make CAR T cells and other biologics to target intracellular proteins as well as just cell surface expressed proteins. So a couple of years ago, Akata and colleagues showed that a TCR mimic binding to HLA were able to mediate rapid and efficient lysis of DIPG target cells. So these two lines here on the left, and this published in 2018, showed this TCR mimic killing DIPG, two different DIPG lines, and then this one on the right is HLA-A2 negative, so unable to present the peptide and sort of serves as a negative control. So we saw this data, and this formed the basis for us making CAR T cells against this target, and again working with our close collaborators at Affinity Bio and their team. So Affinity Bio specialise in making antibodies binding domains to peptide-MHC, which I think, as I said, debunks the dogma that we can only target cell surface expressed proteins. And so we worked with them, and they performed a screen for us, and we pulled out 6 clones of scFv. And I'm just showing the three sort of leading clones here. But so this is an alanine scan of 3 of the 6 clones. So the clones are called A3, 19, and 615. And you can see in this alanine scan that they bind in a similar fashion. A3 and 615 bind in a similar fashion, with clone A19 displaying a slightly different binding footprint, but they lose binding when alanine is substituted at positions 7 and 8. And so we have these scFvs to recognise K27M in context of HLA-A2 and set out to then test these functionally. So again, took a similar approach as what I've just described. So Affinity Bio made the recombinant K27M single chain antibodies, and so we were able to use that reagent and show specific binding to K27M positive target cells. And also then when we cloned, and this is a work actually of Stacey Wang, who's a very talented PhD student in my lab. She's a paediatric oncologist who's doing a PhD on this. And Stacey was able to show really good cell surface expression of the K27M specific CAR on the surface of primary T cells. And then peptide loaded up targets, and was able to demonstrate really good cytotoxicity to K27M HLA-A2 expressing target cells. So originally really excited by this data, and went on to further characterise it. This, I should point out though, this is just a workhorse cell line. This is T2 or TAP deficient target cell lines at this stage, which showed really great promise. And then we were able to show secretion of these various cytokines and chemokines, interferon, TNF, and just in this red, blue, and yellow here, you can just see these different colours, the different clones and their propensity to secrete cytokine and chemokine, as shown here by standard cytokine beta A. And interestingly, so despite really good killing, and we did this with, and I'm showing this data in the true spirit of what I think seminars should be about. They're not all polished and perfect and ready for publication, or if it has been published. This is very much work in progress, and I really would love to hear from anyone who has any insight into this, because despite killing with our scFvs, our CAR T cells, and the BiTEs, bind and kill HLA-A2 K27M expressing targets really well, as shown here, but not the DIPG tumours themselves. So despite even the endogenously mutated K27M mutated line, even when we exogenously peptide pulse the DIPG cells, we really don't see any killing. And you've got this sort of positive control here with the HER2, because these cells express low levels of HER2. So what's going on? So we know that we can make the scFv, we can target peptide-MHC, and we've done this in 293 T cells and T2s and other HLA-A2 tumour cell lines, but not the DIPG. So we've made the functional CAR T and BiTE, and we can kill K27M cells efficiently, but not the DIPG cells. So we tried immunomodulation to enhance peptide presentation, azacitidine. We've tried growing the cells in hypoxic chambers to see if a hypoxic environment would increase peptide efficiency and presentation at the cell surface. We know that DIPG cells are not necessarily resistant to cell death. We know that we can kill them using other targets like HER2, and I've showed you data with HER2. And we also, it was published in that 2018 paper by ICAR T that actually it has the peptide does have a reasonable affinity for the HLA, but perhaps it has too low affinity to displace endogenous peptide and therefore is presented poorly, or other reasons why a TCR mimic can kill K27Ms and CARs don't has to do with the affinity and signaling of those interactions. So yeah, watch this space for that. So in final thoughts, we've created a pipeline in the lab to identify new targets. What's the time? Okay, we've got lots of time for questions, good. So in the final thoughts, we've created a pipeline in the lab to identify new targets. We've generated novel high affinity CAR T cells and other T cell redirection biologics. We've established mouse models of brain cancer, and really strikingly, our novel high affinity EGFRvIII specific CAR T cells are really effective in the clearance of glioblastoma. And this is work that we're all really excited about taking the next steps for. DIPG is proving to be a really difficult tumour, but again, I've showed you that, that we're currently validating some new targets as well, in addition to the K27M project. And then the next steps, we've established all the human T cell systems, and so we're really we are now ready, I should say, to take the next steps in testing our V3 binders in humans. That brings me to the end of my talk, and this is my most important slide because science is not a team sport, and there is an enormous number of people who have contributed to this research program. 4 years ago, when I started my lab, it was all new projects were initiated, and everything was made from scratch, every construct, every cell line, and I really have Ryan Cross to thank deeply for that, and Kathy Watson, who helped me establish my lab. And the work I've presented today here is the PhD project of Beck Abbott and also Stacey Wang, and all members of our lab have contributed, particularly Melinda, who's established the animal models, and none of this would be possible without our important collaborators, particularly Affinity Bio and their team. So Pete, Matt, Ben, Brad and Fiona, Jared Sandow and Proteomics, and Melissa Davis here at WeHi, and our clinical collaborators, the Royal Melbourne Hospital and the Royal Children's Hospital. And we also work with Tony Purcell out at Monash, some of the peptide work, and the Hudson as well. And I want to also, our funders have generously supported this program, and they're really vital to the success of this story. And I really want to thank and acknowledge each of those, particularly Cure Brain Cancer Foundation, Carrie's Beanie to Brain Cancer, Robert Connor Dawes Foundation, Cancer Australia, and the Isabella and Marcus Foundation, who generously support our students. And finally, to patients and their families who generously donate tissue to participate in research to help others. I think that's just an extraordinary decision to have to make. And we thank them for their involvement too. And so with that, I will say thank you very much and open it up to questions.
Thank you, Misty. There are lots of questions.
Great.
So I'll hold mine back
see them,
while we answer others first. So we'll go with the first one. Did CAR T transfer lead to toxicities in mice?
Sorry, say that again.
Did CAR T transfer lead to toxicities in the mice?
Do they transfer toxicity?
Did the CAR Ts being transferred to the mice have toxicities in them?
Oh, no, not at the moment. No, they haven't, but it's worth pointing out that these studies have been done in immunosuppressed mice. So the host mice used in those experiments where we see really striking tumour regression are in NSG mice. So NSG mice don't have a competent immune system, and really it's the involvement of other cell types that can contribute to those dangerous inflammatory cytokine storms that are often seen in patients. So to get around this and to really validate for safety, we're also have made a new mouse so that we can now repeat everything in an immunocompetent setting. So where all of the other tumour microenvironment will be present, and all of those other cells that contribute to inflammation will be present. And so we've made a mouse that will express both human HER2, will also express human EGFRvIII in mice, so they'll be tolerant to that protein, and we can grow those tumours in the brain, and then we'll be able to really answer that question in finer detail.
Very good. Prof. Rosenthal, how and when can you translate this preclinical work in clinical trials for humans?
Yeah, exactly. So we've been chatting to Mark about the phase 0 clinical trial that we want to run at Royal Melbourne Hospital for adult glioblastoma, newly diagnosed glioblastoma patients. And Mark, I think we're getting really close now. we've just the patent. So these two clones have now been patented, and we've just finished writing the paper, and we're doing the safety testing. So the only thing we're waiting for now is we've sent them away to be tested on every normal human tissue, like a tissue bank. And then once we know that they are not recognizing any other antigens and that they are safe, that safety profile comes back, and then we'll be definitely ready for the next steps. So not far away. We should chat.
Excellent.
Okay. Kate said she has 100 patients lined up. Yeah, it'd be brilliant. That'd be great.
From Samuel, did you see signs of immunosuppression that may inhibit the efficacy of the C1 or C2 CAR T cells in your mouse model?
No, we didn't. But again, because we're using those immunodeficient models, it's a hard one to get around. But no, look, we didn't. As I said, we actually just saw complete tumour regression. And when Colleen Darcy was looking in these tumours, she's a neuropathologist at Royal Children's Hospital, and she said she couldn't see one tumour cell present. So there was certainly no immunosuppression at play in these experiments, which was great to see. But whether or not that holds water, I suspect that we will see some when we move to immunocompetent settings, and that's where I think that's where again the data I've showed you is just a monotherapy approach, but also we're keen to try multi-targeting. We have several projects underway looking at a multi-targeting approach as well, that I think would be important to help overcome that immunosuppression that we do expect to see in an immunocompetent setting, such as, for example, genetically engineering CAR T cells to inducibly then secrete bites into the local tumour microenvironment, and Beck's currently working on that at the moment, which would be really cool.
Do you have, Ben Hun, do you have access to, or have you tried CAR T cells in the mouse model that generates endogenous astrocytic lineage tumors such as GFAP, CRE, FLOX, DNF1?
No, is the short answer. That would be really cool. Haven't done it.
Next question from Anonymous. How much longer do the CAR T cell treated mice survive compared to untreated?
Yeah, really great question. Really looking to do those long-term survival experiments now. They're underway. We did have some problems with the mice at sort of, after 6 weeks, them actually coming down with an infection. So we need to repeat that experiment to get those long-term survivals.
Kate with another one. The other line is VMDK mice with SMA 560 immunocompetent.
Yeah, and haven't tried that. Haven't tried that at the moment. So, but now that we know we've got the scFv clones that work, they're functional. We can really now start to sort of tease apart some of these questions in some of these models. It'll be really cool to do.
I'll fire in my question now. As much as I have great faith in Dr. Darcy and looking down a microscope, have you thought about deep sequencing the brains for the C2173 alterations? Not just reliant on IHC, but actually tracking for the DNA itself.
Do you mean looking at the deep sequencing of the T cells? Do you mean, or?
No, for the V3 variant to prove that it's been eradicated.
Okay. No, I haven't thought about that. Yeah, that's a good suggestion. No, we've just looked by histology at the moment. Haven't investigated that any further. They're all fairly recent experiments, actually. They were just done in the last month or two. So no, we haven't, but that's a really good suggestion.
The other thought I had in regards to the K27, when you do your proteomics and you show that your autopsy tumors is totally different than your cell lines,
yeah,
as expected, which is what you see. Have you got any that you've actually taken from autopsy and done in low oxygen environment? You've done the experiments once they've been established in low oxygen, but have you done it straight from autopsy? Because I'd be curious to see if you maintain the protein population in a low oxygen environment.
Yes, that is a good suggestion as well. No, we haven't done that. As you know, they're pretty small and precious and rare samples, and
I'm trying to get you more.
unfortunately, I can't do everything I'd like to do with them just based, on the small amount of tissue. No, we haven't. But to sort of answer that, we haven't done the experiment, but we've got in planning to do the serial sort of dilution as well of looking at how the cells change over time by implanting from mouse to mouse to mouse, and then sort of taking them out and doing proteomics. And so taking, say, patient-derived lines, so implanting the human DIPGs into mice. So we did that, and usually you get about, according to the literature, and there was a big meta-analysis done on this last year, you get about 40% strike rate. So taking some of the human patient cells, implanting them into NSG mice, waiting for the mice to come down with tumours, and normally can take 3 or 4 months, can take 6 months, and then retransplanting those into other mice. Again, we did try that, but unfortunately none of the mice came down and got sick. So, and again, it's just the rare nature of those samples. We haven't done it with DIPG, but it's something that we are planning to do now with glioblastoma because there's a higher availability of the tissue.
Yes, thankfully I don't have a lot of them.
Yeah, exactly, which is a good thing. It's a good thing. I mean, it's rare, but it's just so devastating. So it's a good thing if not so many.
Was there any other questions from anyone else? I'm not seeing anything flashing up. So that'll bring us to the end of today's Monday Lunch live stream webinar. Thank you to everyone involved for the presentations, and the audience for tuning in. And thank you, Misty, for a wonderful presentation.
Thanks, Jordan.